So in some ways, this is like a good cop, bad cop routine, where Chris wants to encourage you that everyone can participate, it's exciting, and I totally agree. But what I want to point out are some of the things in some of my work where I've used Manakin genome quite a bit at this point for some of the papers in that whole 50 genome project. And what I'd like to impress is that there's certain specific things about these newly emerging non-model genomes that we really need to think about in order to be able to use this resource. And everybody here seems like a really excited audience, and I'm sure you're enthused to dive into genome annotation. But I was imagining a hypothetical audience where we'd sit through and maybe fall asleep here, and let's leave that genome annotation to Chris and Moni and Morgan, and I got other things to do. And hopefully we can convince you, if hypothetically you were one of those people, that this is really critical for us as a community to do, especially if you have any interest in using genomic data. So, genome annotation, why does it matter? So, part of the issue is that all of these tools were developed, for mouse, for humans. And so there's the assumption that we have a really complete, well-annotated genome. And that may not be the case. Let's see if I can edit this really quick, because a lot of it's cut off. Yeah, can we edit the screen resolution somehow?
So is it the bottom?
Yeah.
Did you make it in? Did you make it in the other format? Did you make it in square?
PC Mac issues.
Chris, I would just open the presentation to the maximum of the screen and just get rid of all the ribbons at the top and make it bigger.
You mean don't view it in?
Yeah, don't view it in presenter because it's going to probably not show. And then get rid of the notes and the ribbon on top and bars
that should do it.
As we're looking on that, is there an easy answer to the question of how avian genomes differ from mammalian? When I've talked to colleagues about sequencing avian genomes.
Well, they're a lot smaller for one.
Is it just repetitive sequence is lower, or is
they actually have, yeah, they have a lot less repetitive sequence. But one of the findings that we had, we published a paper showing that all avian genomes actually have a huge loss of protein coding sequence as well. And so that actually makes avian genomes very interesting from even a biomedical perspective, because you have like these natural knockouts of even a lot of really critical gene pathways. And somehow birds have solved these issues where these are knockouts of genes that are lethal in mammals. And yet somehow birds have lost these and compensated for it. So yeah, that might be an interesting paper to check out in terms of understanding specific things about avian genomes.
So they're smaller and there's less repetitive stuff. So their assembly is easier and better. So everything else is like genomes are looking pretty good. And then I'd say that like when you compare bird to bird, that organization of genes on chromosomes is more conserved. So all that simply is more highly conserved among birds. So there's less interchromosomal, there's fewer interchromosomal.
So genome annotation, why is this really important for us to get down? So obviously, if you don't have your genome correctly annotated, you're going to have orthologs where you're calling it one gene and it's in fact another gene, or it's one of these models where you have no idea what gene it is. But beyond that, some of these massively expanded gene families that a lot of the people in the room stated in the survey that they're really interested in, like the MHC, color, keratin, feather type genes, these are the trickiest gene families of all, because they're so massively expanded that it can be really difficult for these automated algorithms to figure out what gene is which. Anything that's like a novel gene or a species-specific isoform, those again, these automated methods have no way to deal with these because, it's only in your species. And also transcriptomic analyses, if you want to understand, some of this stuff about expression, you really need to have very accurate gene models. Otherwise, a lot of your data get totally thrown away. And so I'm hoping by the end of this talk, you'll all see why that is the case with some of these things. So Moni's going to be talking a little bit about the genome pipeline, but just to kind of briefly give you an overview, along all the steps, there are generally some really good quantitative ways to assess your data. When you're getting the DNA ready, you can look at gels, spectrophotometry to see how good your DNA quality and extraction were. Once you do the sequencing, you get these scores associated with each read that tell you how confident you can be about any given nucleotide along the sequence. When you're assembling, like Wes has been talking about, you can look at the N50 as well as the contig and scaffold number to get a sense of how complete, how long of stretches of unbroken DNA you're looking at, and also how complete your assembly is relative to the estimated total genome size. But with gene prediction and annotation, the only way you can really assess how good it is to look at it. To look at it in a genome browser and to understand how to look at it. And so in terms of genome prediction, gene prediction strategies really, there are two general and sometimes overlapping methods that are used to do this. And so this is, before we get to annotation, this is just figuring out where in the genome there might be a gene. And so one strategy, there are these ab initio total in silico strategies, where it's basically an algorithm where you feed it gene models from a better annotated species, like zebrafinch, like chicken, and then this algorithm goes through the genome and it says, oh hey, that looks kind of like one of the genes in this set, and oh hey, there's another. And yeah, and we'll talk about some of the issues with that. And then the other is reference guided. So this is also using annotations from something like zebrafinch, something like chicken, but here you're not using like a trained algorithm. Here you're actually aligning the sequence using one of these aligning algorithms to the genome directly.
On that first one where you said it's sort of modeled, is that like a statistical model? I mean, is it assigning probability, or
so let me see if I can flip ahead a little bit. So each of these you can think of as a model. Okay. Each of these predictions you can think of as a model. And so this is a model of, a prediction of where you think your gene is. What do you think the exon intron structure is?
So when you say where you think it is,
where in the genome? So it's the stuff like, where are these exons? Where are these exons not?
Okay.
What is that prediction based on?
Yeah, what's that? I'm just wondering what the information, how that's done analytically.
So basically, you have more vetted predictions from things like chicken, zebrafinch, hopefully, and you're using that information, those sequences, and especially if you have expressed sequences, and you're using that information to, it's literally like an Apple F, a control F. Like when you have a Word document and you want to find the word, dang, and you just want to search through all this text and you want to find where the word dang appears. And hopefully it only appears once, because you want to find orthologs between genes, but sometimes it appears multiple times.
Marian, can you
Yeah, please.
It's also important to know that these ab initio predictors usually are completely based on the actual composition of the DNA. So all of the intrinsic signals that can be picked up from the genome are being picked up through the algorithm, much like a little machine walking through the DNA and then picking out, oh, this is a translation start site, oh, this is a flag that gives you this. And so the structure of the gene will actually be defined by those intrinsic signals that you are able to predict in the absence of any other information, even other gene models. The algorithm will know that those things are genomic elements or genes that are producing proteins, but also for regulatory elements, for repetitive elements, and all of those things can be done in an automated form independently of all the evidence. Then you actually have the alternative to also include evidence in gene models that come from other organisms. And when we say the word model, we don't actually mean a model of a mathematical term or probabilistic. We're talking about a hypothesis. And so each of these genes are basically hypotheses that the algorithms have produced for where most accurately a gene is representing the biology of this particular organism in that particular gene.
So there's the capital T truth of what this gene looks like. And these are all gene model predictions trying to figure out what does that gene actually look like? What's the real intron exon structure?
But the simplest sort of feature sort of thing you could think of, I think, is probably like an open reading frame. Like, so it looks for chunks of DNA sequences that can plausibly be translated into a protein without having a stop codon or something like that. So that would be one feature that an automated algorithm might look for is, can this chunk of DNA sequence, can it actually encode a protein? Because a lot of the genome cannot encode a protein. And so we thought we knew that those cannot be genes.
So does it assign confidence when you're all said and done, when it's like this is there? This my statistical training.
Sure.
So the thing is with the gene models, like I said earlier, there isn't really an easily quantifiable way to determine, that's definitely the gene, or that's not a great prediction. You really do have to look at it. If you have expression data, if you have expression data, you can be more confident, because then you know that was, you sequenced this. This was an express transcript.
Yeah.
But when you're first starting out, you don't have those data. And there are some issues why you can't use those entirely. Cool. And so this is an example of an actual gene. So this is an express sequence transcript. And this is in the zebrafinch genome. And these are several predictions before we had this express transcript in where the gene was. And so these up here are like ab initio gene predictors, where they were fed some sequence, or they were looking for open reading frames, and they were trying to guess where the genes were. And so as you can see, there's some that are like merging multiple genes together, making these kind of read-through sequences. There are others that are predicting genes that may be real, they may not be real. Without expression data, you have no idea. There are lots of other ones that, if you look at an actual alignment of some other genes, and because all of these alignments from different species are going to the same place, that gives you more confidence that this is the actual structure of the gene. A lot of these models are partial models, where they don't pick up all the exons. And in fact, sometimes they incorporate exons that, into other genes where that's not the structure at all. And so if you're using only these automated algorithms, and if you're just trying to team up with some bioinformatician that has no experience with non-model genomes, this is mainly what they're going to do. So this is, again, highlighting why it's really important to think about better strategies for genome prediction and annotation, and then really, diving into it with the manual annotation to curate these models for your genes of interest. So yeah, an all too typical ab initio only pipeline. You make the predictions, you just kind of assume all these wacky predictions are accurate, and then you basically compare them against known sequence databases to try to, based on the sequence similarity, find orthologs. and then you assign orthologs based on some cutoff. And just to show you, a lot of these, they get into great journals. But if you really get into the weeds of what they found, is they predicted 20,000 genes, and only 7,000 of them could really be confidently assigned orthology. And that's their assessment.
What year was that published?
Somewhat recently. I forget, but
A year and a half.
So
yeah,
recently enough to be embarrassing?
Yeah. This is a terrible paper. If you actually look at their gene models, a lot of them are really, really wacky. So
And you did that.
I did some of that. and so this is the issue. It's still kind of the Wild West. We're still trying to figure out better algorithms for doing some of this. And so let me show you just a couple of examples from biology of why this is tricky. And a lot of these statisticians, computer scientists who are inventing these algorithms, they're not biologists by training. So these are some more nuanced examples that you all might be familiar with that they haven't really incorporated into these algorithms yet in in a way that will address these issues. So one is if you have a gene duplication. So as you can probably guess, if you have a gene that's duplicated and now you have 2 copies of the gene, several things can happen. One of the duplicates can take on a brand new function. Different exons can kind of sub-functionalize where parts of it take on new functions, parts of it stay the same. They can specialize in different ways based on selection. Some of them can non-functionalize. They can turn into pseudogenes. But the thing is, it's not necessarily the ortholog that's staying the same. It could be that your duplicate stays the same. So to these algorithms, it looks like the ortholog, this duplicate. But in fact, the orthologous gene is the one that got broken up or that took on a new function. And a lot of these annotated algorithms, they will not catch things like this. Another example is, again, with these massively expanded gene sets. These are the trickiest ones to confidently assess orthology with simple algorithms without manual annotation, because they share high sequence similarity. And so, as you can see, like Augustus, this one predictor, it only finds possibly one thing, and it totally gives up with this stretch. Ensemble, which is generally pretty well curated, they also make a bunch of wacky, kind of broken up models. And even when you're trying to align things to the genome from other known species with better curations, it has a lot of trouble in figuring out what gene is what here. And you can see even the gene direction is, not really known with high confidence. And so this is a feather keratin locus. And another issue with these ab initio only gene annotations is that they're going to miss some of the most important things for us, which are the species-specific genes. So like novel genes that only manakins have, that could be important for, dance and courtship and some of these mannequin-only behaviors. Or you're going to lose some exons that might have changed a lot in mannequins relative to these other species. And these could be, again, these UTRs, these could be really critical for taking a gene and regulating it in a different way. So getting, these like vocal learning type genes into instead thinking about, these mechanical wing noises that they make. And yeah, so these are really critical. And again, without manual curation, a lot of these things are just going to be lost. So again, just to summarize, ab initio, it can capture a lot of these species-unique or highly divergent genes hypothetically, just based on looking at sequence. But as you can see, a lot of times it misses them as well. Yeah, and we'll make sure all these PowerPoints are available for sure, so you don't have to worry about writing all this down. They also make a lot of false positive predictions, combining multiple genes or only finding a handful of exons of the complete gene, it's not enough to establish orthology. And even reference-guided methods where you're taking a gene set and you're aligning it to your gene of interest, I think you get a lot higher confidence doing prediction methods based on that. But it's dependent on using reliable gene orthologs from other species. And even chicken and zebrafinch, it's not completely annotated. Or the annotations are not one-to-one orthologies all the way across yet. This is something I've been trying to do solo in the lab, but if we could, as a community for mannequins, do a lot of this manual curation, we could hypothetically have a better annotated genome than even the zebrafinch. And so yeah, these are more reliable because they're based on other genomes, but they could miss all the mannequin-specific genes as well. Not enough to establish orthology. So now, bad cop's over. So now we're going to talk about some of the things that you can do to rescue some of these things. So one is using hybrid approaches, where you're combining both the in silico ab initio procedures and the reference-guided procedures to make improved gene models. One is using synteny. So show of hands, who's ever heard of synteny? Cool, so a good number of people. So synteny is basically local gene order. And I'm going to show a couple of handful examples that will give you an idea of what that looks like. And then expression data. Again, because when you have expression data, you know that's a real transcript, that's a real mRNA. And so expression data also will help you to confirm that this is the accurate gene. And again, like Wes and other people are saying, you want to have this from as many tissues as possible. So even if you're only into feathers and all you care about are feathers, you really want to have like whole embryo transcriptome is a really important one, and brain is another really important one, because those are two tissues where many, many genes are expressed relative to some of these other tissues. Another one, interestingly, is testes. So here's a vague idea of what synteny is. So these are all the human chromosomes, and this is what it looks like when you map them onto the chicken. So colors that are the same here show you blocks of mostly interrupted genes where they're all in the same position. And this makes sense, right? Thinking about how genomes change over time. These things, they get swapped and they get moved around based on breakpoints. So times when chromosomes cross over at the wrong point, but then it gets passed on. And even between human and chicken, all these millions and millions of years separating the two, even though this might look kind of fragmented, it's kind of incredible how many of these unbroken stretches we have. And so that is a really, really powerful tool. In my opinion, one of the most important, most powerful tools for annotating genomes, for figuring out what is this gene really.
Morgan, is there a way to actually study that without assuming that the human is like the species of comparison that is actually talking about the ancestor?
In fact, you don't want to use human.
Right. Yeah. So talking about some kind of ancestral synteny and how much the ideal species diverges from the ancestral.
The ideal pipeline is actually to look at a couple different species. And it's a trade-off, because in human and mouse, you're going to have the most well-curated annotations.
Yeah, I guess I'm speaking less practically about
again,
how
because there's so much more time, you're going to have things that have broken. This local gene order has been broken. And so you also want to be looking at chicken and zebrafinch generally, and budgergar too, to try to figure out what's the local gene order in a closer related species, where you're going to have much fewer of these crossing over breakpoint duplication issues to worry about. But here's a quick example I picked. FOXP2, everybody's favorite gene. I'm so sick of FOXP2. Here's an example where we just have these predictions, and let's assume none of these have any annotations. So we think there might be a gene here, but we have no idea what gene it is. So let's say we're going to use like a reference-guided approach. We're going to take sequences from a bunch of different species, do our Control F, Apple F, to find where their mapping onto the genome. And we're going to use that to try to figure out what genes these are. And so what you'll see here is based on these different methods, some of them are telling you it's FOXP2. Some of them are telling you it's FOXP1. So which one do you believe? And you'll see this a lot where you're going to get different calls from different methods. And so synteny is basically looking at the neighbors, looking at the local gene order to try to figure out what gene it is. Because again, this local gene order is generally very well conserved, even across all the way down to fish. And so the neighbors here are, I don't know if everybody can read this, but it's like FERM4B, MIDF, IF4E3, and this GPR 173. And so you can just kind of write that on a napkin and keep it in mind. And, everybody loves FOXP2. So let's say we're going to look at like chicken now. This was zebrafinch. So let's say we're going to look at chicken FOXP2. So you can see here, the neighbors don't match. Here we've got TFEC, MIDF, some kind of locus, 1R3A, and a GPR, but with a different number. So here, the local gene order does not match. And so if this were the gene, it would mean that there was a chromosomal breakpoint right here and right here. And that's highly unlikely. Or it could be a duplicate, which is a complicated situation. But now, if we look at the chicken FOXP1 locus, you'll see that it's exactly the same gene. FERM4B, MIDF, I3, GPR. So this, I think, it allows you to very confidently say that that zebrafinch locus was in fact FOXP1. And this is one of the strongest things you can do. And so sometimes you'll get these chromosomal breakpoints that confuse things, but even then, you'll usually have synteny on one side. So this is from a parrot genome paper I'm publishing soon, where we have a gene duplication that happened at one of these breakpoints. And as you can see, if you compare, this is finch, and this is budrigar, and but it's the same in all parrots. If you compare here, you find two PSMD 6s. And so which one is the ortholog? And it turns out because of the duplicate, they're both kind of the ortholog. You can't establish clear orthology. It's one-to-many orthology is what you'd call this. And so one of them has synteny on one side, the same genes, and then it's different on the other side. And one of them, prickle atom, is by the other copy. So that shows you there was a breakpoint, but synteny was conserved on either side. Duplicates are hard, but not impossible. So trying to blaze through the end here, but expression data is really where it's at to validate predictions. Again, expression data doesn't give you annotations, but allows you to strongly improve the predictions. And so this helps you validate the models that you predicted earlier. It allows you to improve and extend gene models, and also to discover novel genes that none of those techniques were able to pick up at all.
So I just want to go back to your last example. So can you, based on this particular example that you gave where you think the breakpoint is, can you assert which of the duplicates is maybe the functionally transcribed gene?
Yeah, you can. Well, but you use expression data to figure out whether they're transcribed.
Right.
But to establish which one is closest to the ortholog, you do one of these alignments. And that can tell you like which of those cases earlier I was talking about, which of those cases occurred. And so in this case, one of them got truncated, and so it could be acting as like a dominant negative where it's regulating the main one. And the other one looks a lot like the ortholog. But a lot of these cases can happen with duplication, any of them really. Depending on the gene, depending on why this was selected for and carried on in this population. So here's an example of these RNA-seq short reads. So these are the data you're going to be collecting when you're doing transcriptome work. This is what it looks like when you're actually looking at them in the browser. And so here's like an incomplete gene model. So this is one of these models where you're predicting there's a gene there, and here are the reads. And so here's 5', here's 3', and what you can see is a lot of these exons are strongly supported from these different tissues. There's sometimes where you'll see that a tissue has no reads, gives you some interesting information about where these transcripts are expressed, more or less. But then you see, after you go from the end of your gene model, it continues. And so if you're just using one of these automated RNA-seq pipelines, none of these reads get counted. And so these are the only reads that you'll have to give you information about what genes are expressed. And another issue here is that if you have two really similar genes, the best pipelines will throw away reads that map to both, because you can't confidently say that it came from this gene or that gene. And so the 3' UTR is where you're going to have a lot of reads that are really specific to your transcript. And we all can figure out why this is. It's because these exons, this coding sequence, is strongly conserved. There's strong kind of stabilizing selection usually to keep these coding sequences conserved. But these UTRs, these non-coding exons, they can change a lot, because this is how you change where the thing's expressed, where it's regulated. And so this is where you're going to get, for a lot of times, all your reads. And if you have incomplete gene models, they're not going to be counted. And so this is why it's really critical for your genes of interest to look at the transcriptome data like this, and then Moni's going to show you how you then extend the model so that you can capture all of your expression data.
So most studies just throw that stuff away,
Yeah.
including everything I've ever done. We don't look at that. We don't have those UTRs in. Those reads that fall outside of the current gene models for zebrafinch, most people don't do anything with those. So you throw away a chunk of your data that's totally useful.
Totally useful. And especially, again, for these like feather keratin genes. All of these reads, the exonic reads, or many of them, are going to get thrown away, because you can't confidently say which of your keratins they came from. And then if you don't curate your model, all the reads that are left over from these non-coding exons, you're not going to count. So this is, again, why it's really critical to do this manual annotation.
I think you were alluding to this the other night with me when I was asking you some questions about if your goal is to use RNA-seq data for gene expression quantification. Is this what you're talking about?
This is what I'm talking about, yeah. And so this is why quantification of RNA-seq data is still somewhat an issue relative to things like qPCR, which are more precise in a lot of ways for this reason. Because here you're throwing out some of the data. You're throwing out the multi-mapped reads. We can talk about this later in more detail.
Well, is it going to throw out all of that?
No. I mean, again, if you don't curate it, it's going to throw out the multi-mapped reads, and you're not going to see these reads because you didn't curate your model. Because these are the ones that are going to be left over that are more specific generally to your gene of interest. But again, if you don't have a good model, these algorithms aren't going to see those reads as being part of the gene.
And it sort of depends partly on, again, like Wes mentioned earlier, this varies from genes. Some genes are going to be really bad. Other genes are going to be pretty close to right.
Yeah.
So
it kind of depends.
Yeah. And it also depends on how highly expressed the genes are. So if you have a lowly expressed gene that you care about,
like transcription factors,
you don't want to be throwing out reads. But if your gene is expressed pretty highly, like this one is, I mean, you will still be able to quantify it with that many reads mapping to the exon. So, there's some,
So naive question. So on RNA-seq will recover UTRs upstream?
Not always.
Not all.
It will help you in some cases, but it depends on the coverage. And it depends on the, sometimes it will, but not always.
So Wes brought up the question the other day of, do we want one deep coverage genome or many low coverage genomes? And based on what I've shown you, I think that having one deep coverage genome, at least to start with, is critical. having one where you've sequenced it in great depth using, very high coverage PacBio sequencing, so that you can use synteny, like I was showing you. You can have long stretches of unbroken reads, long unbroken contigs, where you can look at that gene order, and you can establish orthology. And also, a lot of these non-coding regions, these 3' these 5' UTRs, a lot of times when you do, and this is true for the Manakin right now, when you look at the actual genome, those parts are not sequenced a lot of the time. And there are various reasons for this, but yeah, but again, these critical regions, if you don't have deep enough coverage, you're not gonna have them. And you're gonna have a lot harder time establishing orthology.
What's your number of reads that you're calling deep and shallow?
This is something to talk to Wes about, but generally PacBio, I think like 50 to 70.
Deep, in my opinion.
Yeah.
Per birth.
Yeah.
On 1 million genomes, maybe 80, 90 X.
Yeah.
And the 48 genomes are of variable
Variable
They're all
quality.
Illumina, and they're all about 100X roughly.
Yeah.
And so
Some
you
are
said
better
that
than
there's
others.
specific gaps in the current Manakin genome that
Yeah.
how would
Lots
they
of
be addressed?
Deeper coverage. Deeper coverage sequencing.
So that means sequencing
More
more
sequencing.
different individuals or the same individual?
Ideally the same individual.
I'm sorry?
Yeah.
Well,
Well, I'm hearing two different things and not typically.
Well, if
So,
you want to do a single genome, then you want to do the same.
so how do we solve this problem now that it exists? Because we don't have access to the same individual, right?
No, we
Yeah,
do.
we
We
do.
just resequence a lot.
Yeah,
PacBio.
Yeah.
And we're adding that to it right now and seeing how that helps.
Mhm. So yeah, we're adding PacBio, but you want as deep coverage PacBio as possible.
What was the coverage?
20.
Yeah.
The PacBio that we got should be in excess of 10. We were shooting for 10x, and we got good recovery on all of those smart cells. So it's going to be in excess of 10x, but probably not 20x. And that's based on recommendations of people that are focused on doing hybrid assemblies. So we'll see. Again, they do a lot of tweaking when they're doing the assemblies themselves. You just use their software that you can download. It may not work as well.
Mm-hmm.
We're in the middle of trying it right now. Paul and Rebecca have run it once with the CAN software for 5 days and then crash, right? So we'll see. We were hoping to actually have the first pass hybrid assembly done by this meeting, but it crashed.
And the second run's going right now. I think about 24 hours. So.
But again, we have the same individual, so yeah, we have the same individual. So hypothetically, we could increase the coverage, sequence it with even more depth, and that would help all of us.
We know where the coders live, so they live in the Washington area. Their hands on them.
So this is my last slide. So an improved pipeline, you use a hybrid, kind of reference guided and ab initio gene predictions. You get expression data from many different tissues in order to validate them, in order to extend and improve these models, look at different isoforms, different splice variants, as well as your species-specific genes that may or may not be picked up by these methods. And then finally, you're going to do the manual annotation. You're going to help out with this effort, and you're going to use synteny, this local gene order, to verify what your genes are. And using synteny, it's largely manual for now, but I'm developing some algorithms. Other people are looking into ways that we can make this more annotated, so that hopefully, we can get away with doing a little less of this, deeply manual effort for right now.
That's it.